maximum, but then before that, like before 48 hours, you will eat the pill, but then after, like before 48 hours. So what if you eat the next day after intercourse, and like in the first day only fertilization happens, then the use of that will be useless.
Yes, I think Fatima's questions are like those questions which make, she's asking a very simple concept, but in a way where it's very, very difficult. It makes the scenario difficult. Now see, there are two things, Fatima. If someone is already fearing pregnancy on a given day after an unprotected sexual activity, then the next day, chances of her again going to commit the same mistake is very less, right? In a sane mind. So technically, it's already not the minimum limit is already not going to be tested. But still, because you have asked, yes, even in the next day, once you have taken the pill, on the next day, it does not matters what is the minimum limit. Okay. But to answer your question, it has to be a few hours, like at least 4 to 5 hours for the sperm to reach the uterus or the place where fertilization can happen. 48 hours is actually the maximum time for which a sperm can survive in the female tract. That is what I'm saying. You understand, Fatima? It's not the time taken by the sperm to reach the ovum all the time. Even if a sperm get let's say derailed, or let's get lost somewhere, but if it is alive for 48 hours, it can again find its way to the ovum. That is what I'm trying to say. Okay.
Yes, sir.
Yes, quickly, I was telling something about the tattoos, right? You would like to hear, people?
Yes, sir.
Yes, sir.
An interesting research study that shows, when we put ink in to make a tattoo, that ink goes inside a cell, right? Yes or no?
Sir, it
Yes,
does.
sir.
We are putting the ink in the cell or in between the interstitial tissue, right? But people started thinking that why does not tattoo disappears after few years, because these cells will constantly keep dying and replacing. Yeah, but tattoo stays there. Like even from birth to death, the tattoo will be there, because interestingly, the replacement of these cells happen in a manner where when the first cell is about to die, the second cell comes in its place exactly at the right moment and keeps the ink exactly there. Because it's not knowing that it is ink and it is not useful or it is harmful or something. It just knows that whatever is wherever it is, it has to be kept there. So it's just like you are holding a black thing in your hand, you are a cell, and then I replaced Fatima for the duty. Fatima handed that over to me. Now I am holding that black thing in the hand, but exactly at the right place. Interesting, no?
So is it
But
like that
then they
for
do,
cells who don't have a tattoo as well?
sir, but they do fade away in due time.
Exactly my question.
Like,
They do. Yes, they fade away because some amount of the ink gets leaks, and the upper layer keeps on it keeps on wiping off. So it becomes faded, but it stays exactly there, right? It's not like you made a tattoo here and after some time it will come here, or it will grow big or small.
then, if sir, what about the tattoo removers? Sir, are they killing those cells which are
You are
holding the ink?
you are removing the ink. You are removing the color of the ink. We can continue it in the next class, okay? Because I have Reem has joined and I need to take her class. So everyone, we will continue this in the next class and then we'll finish the chapter. Okay?
Okay, sir.
Thank you, sir.
So thank you,
Thank you,
sir.
sir. Bye.
Good evening, Reem. You're there?
Yes, sir. Good evening.
Yes. Sorry to keep you waiting for some time. It was the last batch. They were having a discussion. Okay.
It's fine.
Yes. How are you? You were not there in the last class.
Sir, I was out.
Yeah, you missed some very interesting discussion on genetics. Were you there in the first class of genetics? Yes, you were there.
Yes.
Yes. Right. Correct. So you missed certain aspects of the last class. So what should we do? Should I give you a recap of chapter 3, chapter 2, or of the last class? What do you want?
Last class.
Genetics.
Yeah.
Okay. So can you see the screen?
Yes.
I think Tiha will also join. But let's continue. If she will join, we'll come to some concepts of plant, okay? If we have time.
Okay, sir.
Will be so till where did you attended? Last to last class?
Till the cross.
Sorry?
Till the cross.
Process, right? So
Yeah.
did we do incomplete dominance when you were there?
No.
No. Okay, cool. Okay, so in last to last class when you were there, Reem, you must be remembering that we studied law of dominance, right?
Yes.
Law of dominance tells us that there is one allele which is dominant over the other allele whenever they are present in a heterozygous condition. Yes. And we also did many type of process by changing different parents. If the parent is pure line, then it is what NCRT tells you about. But parents can be others also. Like in entrance exams, they can say a hybrid tall is crossing with a hybrid, another hybrid tall. Things like that. So all this is clear, right? The phenotype, genotype.
Yes, sir.
Okay, so after that, in the last class, we did some exceptions of law of dominance. Remember, I told you that law of dominance is not a universal law. It is not always followed. When Mendel gave the law, because one limitation for Mendel was a brilliant botanist and a brilliant scientist, but one loophole in his experiments was he never tested any other organism. He did everything on peas, gave the laws, and just thought that how peas will behave, every other organism will behave the same. But that was not true. After Mendel gave the laws and after he died, people started figuring out that the laws of Mendel does not hold true for other species of plants and animals. So there was one concept called incomplete dominance that came in. Okay. Now, incomplete dominance simply means that the two alleles in a heterozygous condition, one was completely dominant over the other, right?
Yes.
If I'm crossing capital T with small t, it always has to be tall. Small t has no say, okay, no power. But in incomplete dominance, the dominant allele was not able to exert a complete dominancy over the recessive allele. So there was blending of traits. Blending means mix up. You will see that the characters are not purely coming from the dominant trait, but they are a mixture of dominant and the recessive both. You understand?
Yes, sir.
In this case also, recessive is also not expressing, okay, but it is slightly expressing, or it is not allowing the dominant trait to express completely. That is the difference. Do you understand, Reem?
Yes, sir.
One example is snapdragon plant, the flower of it. So there is a plant called snapdragon, and the snapdragon flower has a very unusual genetics. People figured out that the plant has two type of flowers. When it is pure line, either it is red or white. Capital R will give rise to homozygous pure line red. Okay, see, look here. I've already. So look here. If it is capital R, it will give pure line red, just like a pure line tall. Makes sense. Okay. If it is smaller,
Yes, sir.
recessive, then it is pure line white. Makes sense. In normal case, if you would have crossed this, a pure line red with a pure line white, in the next generation, all the progeny should be red, right? Hybrid red. Yes or no?
Yes.
Instead, in this case, what happens? In the next generation, in F1, all the progenies are pink because all are hybrid. But instead of being hybrid red, they are pink because when red and white mixes up, what becomes?
Pink. Sir, in Mendel's law, there's no such thing as blending, right?
Yes. That is why it is called exception of the law of dominance. This is an exception of Mendel's law.
Okay.
Okay. Mendel's law are pure. Mendel was not aware of blending or incomplete dominance. So he said, whenever in a heterozygous condition, two alleles are present, only one will express purely. The other will be recessive and masked. But we figured out that in Snapdragon, that is not being followed. A red flower crossed with a white flower gives a pink flower in the first generation. So there is some blending happening, right? So this is
Yes.
first exception. And because of this, what happens? The genotypic and the phenotypic ratio changes. How? Let's see. So before I give you answer, why don't you do it? So you understood that when these two will be crossed, all will be heterozygous in the F1, right?
Yes, sir.
Now, please right now make a self cross of F1. Make
Okay.
the cross and tell me what will be the genotype and phenotype. So this is F1. Tell me what will happen in F2. Are you doing it?
Yes, sir, I'm doing it.
Can I just get myself water, Reem?
Yes, go ahead.
Let me know when you're done, Reem.
Sir, the genotype is 1:2:1.
Okay. And what is the phenotype?
That is also 1:2:1.
Are you sure?
Yes,
Correct.
that's correct.
Yes, that's correct. Because you understood this phenotype should have been 3:1, right? Normally.
Yeah,
Under
but since blending is there.
yes. But because it's blending, now there are 3 phenotypes possible. Red, pink, and white.
Yeah.
So the genotype is not affected by the incomplete dominance, right now.
Okay.
Incomplete dominance, because genotype normally also was 1:2:1, right? So genotype is not affected by incomplete dominance. The blending only happens in characters and hence phenotype is affected. The blending only happens in character and only phenotype is affected. Okay, Reem?
Okay, sir.
Understood, right? No problem. Okay. The other thing we did was the other exception. So one exception is incomplete dominance. The other exception to law of dominance is codominance. Now, what does the word codominance means? It simply means that there are more than one alleles and two alleles are equally dominant over each other. No one is recessive, no one is dominant when they are present together. They both are co-dominant. Is it clear?
Yes, sir.
Both are equally strong. So this also violates the law of dominance, because law of dominance says whenever in a heterozygous condition, one should be dominant purely. So codominance happens in our blood group. You know about the blood group of humans, ABO blood grouping.
Yeah.
So if I say that, you tell me first, if I say that my blood is O positive, what does it mean?
It means that your parents had.
No, forget about parents. We have not come to parent generation. Tell me about my blood type. If O positive is my blood type, what does it mean with respect to the antigens of my blood?
That it is very rare and that you're a universal donor.
O positive is the most common blood.
O negative is rare.
Yes. The rarest is AB negative. AB negative is rarest. Less than 1% of the world population is AB negative. The rarest blood group. O positive is the most common. 42% of Asians are O positive, including me myself. Okay, so this is the most common, this is the most rare. This is O positive The O blood group is called to be universal donor, and the universal donor is O, um, and the universal acceptor is AB. That's all there. But what does, there are three, it means that there are three antigens on our RBC, okay, on the blood, which defines the blood group. Antigen A, okay, antigen B, and no antigen. O is not an antigen. O means no antigen. You understand?
Yes, sir.
So O is absence of both A and B. But there are three alleles of the for the gene. One allele gives rise to A, sugar A. So this A is a sugar which act as an antigen on the surface. So sugar A is coded by allele A for the same of the same gene. There is another version called allele B, which codes for sugar B. There is a third allele which does not code for any sugar. You understand? But there is a gene. It does not code for any sugar. It's not like there is no gene, because it is not possible that someone has no gene for the blood antigen, right? It's not
Yeah.
possible that someone does not have a gene for eye color. There has to be a gene, right? Whether that gene is producing pigment to give a color or not is a different thing, but there has to be a gene. Yes or no? For the character.
Yes.
So there is a gene, there is an allele, but it does not code for any sugar, any antigen. So if someone's blood group is A, there are two possibilities. Either one parent give allele A, right? IA, and the other parent also give allele A. Yes? So in
Yes.
a homozygous condition, A will be expressed. Yes or no?
Yes.
The next condition is one parent give allele A, the other parent give allele third, 3 or O. So allele O is anyways not coding for any antigen, right? So the only coding will happen is from A. So obviously the blood group will be A, right?
Yes. Sir, in textbook it is small I.
Yeah, small I is for, I will come to small I. Small I is for allele. I am talking about here about the inheritance.
Okay.
Right? Capital I with A, it means the inheritance, the chromosome, the gene that has come down from parents. The allele is called this. Okay. So there is one allele of A, allele of B, and there is small I allele, which is for O. It does not code for any sugar, no antigen. But there is allele. Is it clear, Neem?
Yes, sir.
Textbooks also tell you that in case of this heterozygous condition, so see, this is a heterozygous condition, no? Technically, though, it is a heterozygous condition, no? There are two
Yes.
alleles present. So textbooks say that A is dominant over O, which is not incorrect also, because yeah, A is expressed, yes. But you can understand why O cannot express, because O does not code for anything, any sugar. You understand?
Yes, sir.
Technically, there is only one thing to be dominant, and that dominates, yes. If someone's blood group is B, then also it has been seen that either it has to be a homozygous B, or it has to be a B and O heterozygous. So again, O will be recessive and B will be dominant over O. Yes. So when these two
Yes.
combinations are present, the law of dominancy is followed. Now there is a reason behind it, because O is not contesting at all. If you and me are running in a race and I do not start from the starting line, then obviously you are going to win the race. Yes.
Yes.
Still you can say that I won the race because there were two participants. So here also there are two participants. There are two alleles. One is being expressed over the other. So it is law of dominance fulfilled. Yeah. But you know the internal reason now. So in this case, there is law of dominance. But in case of a AB blood group, when one parent,
It is co-dominant.
yes, when one parent give allele for A, the other parent give allele for B, these two alleles cannot decide which one is dominant. They are both co-dominant, and even in each other's presence, they both express with equal strength. And the blood group is AB. You understand?
Yes, sir.
In AB blood group, there is a co-dominancy of antigen A with antigen B, allele A with allele B. And this is also a heterozygous state. Yes or no?
Yes.
For someone to have a blood group O, the only possibility is both the parents gave O, the allele, the small I allele, this allele. So this is the gene, capital I. It has three alleles, IA, IB, and small I. So if both the parents will give small I allele, then only this combination is possible. So for me, I received recessive allele, allele O from both my parents. This is what you were answering in the first place, right?
Yeah.
But is it possible that, tell me, if my blood group is O positive, is it possible that my parents' blood group could be A or B?
Yeah, it is possible.
How?
Because small I is also there.
So
Maybe due to that.
my mother could be this, right?
Yeah.
Or my mother could be this also. And she gave me this one in a gamete, right? My
Yes.
sister also can be either A or B, and he gave me this one in the allele. So it can be. But is it possible that any one of my parent had a AB blood group?
No, I don't think so.
Yes, this is not possible. If the kid has blood group O, none of the parent can have blood group AB, because if this is happening, then the kid is either adopted or not biological. And these type of questions are asked in the entrance. Okay.
Okay, sir.
If my mother had AB, either she could give me A or B, right?
Yes.
In of the cases, I would never be having a O group. Yes.
Yes.
Yes. So parents can have A or B, but parents cannot have AB for a O group kid. You understand?
Yes, sir.
Do you now understand why biologically O is most prominent and AB is the very less? Because
Yeah,
we
yeah,
come from
I do.
AB parents. Yes.
Yeah.
They can also come from A and B parents, but O can come from both, all three, A, B, and O. From here,
Yeah, but AB can't.
here also O can come, and here too obviously O can come. Make sense, Reem?
Yeah.
Very good. So in blood group, we have codominancy, which is again a exception of
Mendel's law.
law of dominance. Now I gave some questions. I'm giving the same question to you. Just don't look at the screen. Write down the question and solve this before others join. This was the last thing we did. Oh, no, we also did law of independent assortment. But quickly do this one question. Write down the question. Parent one had a A blood group, and parent 2 had a B blood group. Okay.
Okay, sir.
First parent had a A blood group, the second parent had a B blood group, and both of them had kids. So some kids, write down, some kids had O blood group. Okay. Now there are three parts to this question. First part, is this possible biologically? You have to say yes or no.
Yes.
Second part, if it is possible, what are the genotypes of parent 1 and parent B?
It is IA and IO.
Yes.
And for parent B, it is IB and IO.
Perfect. It can never be IA, right? For
Yeah.
and also it can never be IB for parent B. And third, you have to tell me what is the genotypic and phenotypic ratio of F1. This you have to make a cross.
Yeah, I'll make one.
Yeah.
Sir, it is 1:1, 1:1, 1:1.
Perfect. both genotypic and phenotypic ratio will be 1:1:1:.
We can write it as 1, right?
No, there's a difference between that. We can write it as 1 in this case. Let me tell you the difference between 1 and 1:1:1:1. It is still a ratio, right? Because
Yeah.
4 possible different genotypes, and for each there is one progeny. You understand?
Yeah, so we can't write it as 1.
Yes, and there are 4 possible phenotypes, and there are each progeny for one phenotype. Yes, so it is.
Yeah.
It will not be a ratio in this case. Let me go up a little. We did some pure line cross, right?
Yeah, and the ratio was only 1.
Yes, in this case. In this case, where a pure line tall is crossed with a pure line short, in the next generation, there is no ratio.
Because everyone is tall.
Yes, all the 4 is same genotype, and all the 4 are tall. So there is no ratio. Then we can write it as 1. No ratio. You understood the difference, Lee?
Yeah. Yes.
Very good. Perfect. Now, before others join, let me quickly also tell you about, we started within the next class with, so all of this, the law of dominance is being followed in the two, actually single gene inheritance. It's called single gene inheritance. Then we went to the second law, which is called law of segregation. Law of segregation is the only law which is universal. It has no exception, okay? And it is known as law of purity of gametes. Actually, throughout this all the process that we did, we followed law of segregation. How? Every time when we had a genotype and we were making gametes, each allele was segregating from another allele independently. Yes?
Yes.
Every time when we were making gametes, these gametes were pure. If this is IBIO, there is a concept of dominancy. If this is red with white, there is a concept of incomplete dominancy. If there is A with V, there is a concept of codominancy. But when again we are making gametes from even these cases, gametes are pure. No incomplete, no blending, nothing, right?
Yeah.
We always, otherwise we were not able to make these crosses at all. So every time, every 2 alleles, when they are present together, they can do all sorts of things: codominance, incomplete dominance, complete dominance, anything. But the moment when they have to make gametes, they all behave the same. They have to segregate independent of each other and go into different gametes. Yes or no?
So that means they are homozygous.
That means they are,
Homozygous.
no, homozygous is this state. This state is homozygous. But even homozygous conditions segregate independently. See, this went there and this went there. You understand?
Yeah, yeah, I get it.
Yes. So for making gametes, alleles have to be pure all the time. That is the rule. And that is called law of segregation, that gamete alleles always segregate independently of each other to form gametes and to keep the gametes pure. So it's called law of purity of gametes, and it is a universal law without any exception. Okay.
Okay, so
So that's it about law of segregation, nothing more. Then we come to law of independent assortment. From here onwards, the third law is for 2 gene inheritance. So for this, till this time, we were only talking about one gene, like gene for tallness with 2 alleles, gene for blood group with 3 alleles, gene for flower color with 2 alleles. Yes or no?
yes.
Gene was single. Now we are talking about law of independent assortment, which only works or holds true for 2 genes. For example, round is for seed shape and yellow is for seed color. So there are 2 genes here, right? One is for seed shape and the other is for seed color. Yes or no?
Yes.
So if I make a cross, let's say capital R is for pure line round. Okay. and capital Y is for pure line yellow. Okay.
Okay.
And there are two genes which are now going together. This is exactly what happens when genes are inherited. They are all inherited together. It's not like one gene is inherited at one time. Just for the sake of understanding certain laws, we do it, we read it like that. So now we are into multigenic inheritance. This is two. In NCERT, polygenic or polygenic inheritance is not discussed in detail, but I will teach you for entrances. So from one, we have now moved to two genes. So this is round and yellow, pure line round and pure line yellow. What is this? Pure line wrinkled, smaller, and
pure line green.
Okay. So if I cross this thing with this thing, now will you please do the cross yourself?
I'll try.
Yes. Try. What will happen? Are you able to do the cross?
Okay, sir.
Are you able to do it, Reem?
Yes, sir. It's complicated, but I can do it.
Now, do you understand now that, see, the rules are same. Find the genotype of the parent, which is already given to you here.
But it's very long.
Make the gametes, yes. And then make the Punnett square. Hello, Minashi, how are you?
Good evening.
Good evening, good evening. You have just joined. I was just giving some extra time to read. So don't worry, okay? Don't think you just joined. I'm just recapping, okay, Minashi, what we did in the last class. So it's a recap, which is a help for Reem, okay? So just be, yeah. Let others join. Where are others?
Sir, the genotype is 1:2, 2:2, 2:4, 4:1, 1:2, 2:1, then 1:2, and then 2:1.
Yes, so for this, I just asked you in the F1, okay? So
I did the whole cross.
you did the whole, right? So in the F1, all will be round and yellow.
Yeah.
Correct? Then you also self-crossed. I understood
Yeah.
a lot of time, but I understood. So in self-cross, you made, so there are 4 possible gametes for each parent, right?
Yes.
There will be a 16 chambered Punnett square, right?
Yeah.
This is the Is this your genotypic ratio?
Yes.
2:1:2:4 2:1:2:1.
Yeah.
This is your phenotypic ratio, 9:3.
Yeah. 9:3:3:1.
Perfect. Then it's okay. And in today's class, I gave Minashi. Now, where are you? Did you go back and read about linkage, recombination, mutations? So today we are going to discuss the exception of the third law, which is law of independent assortment. You understood the law, Reem?
Yes, sir.
Now let's look at their exceptions. Okay. So can I just wait for 1 more minute for others to join if they are joining? Otherwise, we'll start. Okay.
Okay, sir.
I also gave a homework in the meantime. You can write down this homework. Do it after the class today yourself, Reem.
Okay, sir.
You have to find genotype and phenotypic ratio. Okay, so Minashi and Reem. Let's start. Minashi, are there?
Yes, sir.
So as I told you in the last class, there is only one law out of three which is universally accepted, which is known as law of segregation of gametes, the second law. There are exceptions to the first law that we have already studied. Now there are also exceptions to the third law, which we will study now. So what does the third law says basically? Minashi, can you give a recap for the third law for me and Reem? What does it say?
It states that two pairs of characters are combined, and when the segregations, the one pair of character is independent over the segregation of other pair.
Yes. So Reem, did you understand? I did not tell you the law till now. The law is,
Kind of.
yes, the law is when two characters, now we are talking about two different characters, not alleles, okay? When two characters are present together, when they are inherited to the next generation, law of independent assortment says that both these two characters assort independent of each other. Any combination can go in the offspring. And that I will prove. Look here. What were the parents when you started this cross? One parent was round and yellow.
Yeah. Round, yellow, and then wrinkled, then
Wrinkled,
green.
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 Now, if wrinkled character, like the character for color, seed shape, and the character for seed color, if they were not able to go independent of each other, then in the next generation, when everyone is round and yellow hybrid, then in F2 generation, the combinations which are different from the parents will not have been formed, right? If these
Yes.
are linked together, let's say wherever Reem goes, Minashi follows. Like they are best of friends, always going everywhere together. So the chances of finding Reem in a separate place and Minashi in a separate place is very difficult, right?
Yes.
But if both of you are one each character, but you going wherever you want is independent of each other. You can also happen to be at the same party, but if someone sees you together at the same party, that does not ensure that every time you will be together, right? You can independently assort and go to wherever you want, right? To make any combination.
Yes.
That is called law of independent assortment of characters, of genes. Okay, is it clear? And that is true. That was true for pea plant because by crossing round and yellow parents, we also got round and green progenies. We also got wrinkled and green progenies. And we also got wrinkled and yellow progenies. And of course, round and yellow progenies, so we will get. You understand? All 4 different combinations are possible. 9 is
Yes, sir.
So this says that yes, law of independent assortment is correct. Two characters are capable of independently assorting. when it goes to the next generation. They are not linked together. But again, when Mendel did that, he did not know that there are certain characters, there are certain genes which are really linked to each other. So wherever one gene will go, the chances of other gene going there will be more. And if the linkage is 100% linkage, then every time these two genes will be inherited together. So either an embryo, a progeny will get both or will get none. You understand?
Yes, sir.
This is a violation of law of independent assortment. And that's why it is an exception to the law. Is it clear?
Yes, sir.
Now let's understand it in detail. So write down, let's start with some writings. Write down, law of independent assortment. Law of independent assortment states that when two pairs of characters are combined in a hybrid together, when two pairs of characters are combined in a hybrid, the segregation of one pair or the assortment of one pair, you can say anything. The sorting, segregation, or assortment things are same. The segregation of one pair of character is independent of the other pair of character. Is it clear?
Yes, sir.
Next line. And therefore, all possible combinations can be expressed. And therefore, all possible combinations can be expressed in the second generation, F2 generation. In the second generation. Now, to understand the linkage and recombination, first we have to understand, you both understood the law of segregation, sorry, law of independent assortment.
Yes, sir.
Yes, sir.
Now, before going into the linkage, let me teach you about something called, write down, the chromosomal theory of inheritance. A very simple but very powerful theory. Question is asked, who gave this theory? Mendel, write down. Mendel gave this theory, but it was not appreciated till his time. So chromosomal theory of inheritance. What does it says is all the inheritance of all the genes now happens in the form of chromosomes. It is a very simple thing. We all know of it right now, but at that point of time, it was something revolutionary. So write down. Mendel gave chromosomal theory of inheritance, and in boards, in school exams, sometimes they ask, why did write down this question, why did chromosomal theory of inheritance remained unrecognized till the beginning of the 20th century? Till the beginning of the 20th century, which means 1900. Okay. The answer for this is, first thing, earlier there was no internet, right? There was no papers being published and being circulated through internet. So a scientist at one corner of the world, when used to figure out something, it could take years for that work to reach other place, the other continent. You understand?
Yes.
Yes. And second thing, in previous time it was possible. Sometimes you will also read that to find this thing, two independent scientists were working at very, unrelated regions. Like one was in America, one was in Europe. They both were working on the same thing, and they both found the same type of result during the same time. So both of them are credited independently. You understand? Because both of them were not aware of each other's work. And that used to happen a lot early because the communication was less. Nowadays, whenever someone finds something, it's on internet, right? And the next day, no one will start working on that again. You understand? Because it's already been done. So you do something better, more. Is it clear, everyone?
Yes, sir.
First factor was, write down, communication was limited. First factor, communication was limited and research work was not widely published or publicized. Publicity was not there. Communication was limited and research work was not widely publicized. Okay. And second problem with Mendel was that he thought his concept of genes, which he actually called factors. So gene as a term was not there when Mendel was doing this experiment. So Mendel used to call all of these factors. So there is a factor for tallness, there is a factor for shortness, there is a factor for this color. You understand? But his concept was that these things are discrete, they are non-changeable, non-negotiable, and they cannot blend. For example, there is a 1 rupee coin and a 2 rupee coin. Now, if you have to transfer it, you cannot blend it and make it a 3 rupee coin, right?
Yes.
You can only transfer it as a 1 rupee coin and a 2 rupee coin, or together as a 1 and a 2 rupee coin, which works like a 3 rupee in total credit. Do you understand? But it will never blend.
Yes, sir.
So when Mendel said, let's see, I believe that all these factors are discrete and I believe that they can never blend, then the other scientists present at that time did not accepted it, because some of them already had started getting the hint of incomplete dominance. Okay, but they could not prove it at that time. The work was ongoing. But write down the second reason for Mendel's chromosomal theory of inheritance to remain unrecognized was, write down, Mendel's concept of genes, in brackets write down factors. He called it factors. This is a neat question. What did Mendel call the units of inheritance? Genes, alleles, factors, sports. He called it factors. Okay. So write down, Mendel's concept of genes was that of a stable and discrete unit. A stable and discrete unit that controlled all the characters. That controlled all the characters and its expression. According to Mendel, alleles never blend with each other. Okay. And this point was not accepted by other scientists. Can you give me one very common reason why it will not be accepted? Like their thought was also logical, hence they cannot prove or disprove it. Mendel proved it through pea plant. He said, see, the moment I cross a tall with a short, all are tall. Why is there no short? Because they never blend. But then someone could have said, have you looked at the skin color for humans? Is it either black or white? Are there only two shades in human skin color?
No, there are several.
There is tone, right? A gradation.
Yeah.
Here there are fair 1, fair 2, fair 3. You get that tone for foundation and concealers. You must be better.
Yeah.
So you get, there are hundreds of numbers, each signifying a tone.
Yeah.
So they said that if it is never blending, then how about human skin color? How about human height? Is it like everyone will be 6?
No, they're medium, like average height.
No, but even in that, every human's height can vary by millimeters and centimeters. Someone can be 5 foot 6 inch, someone can be 5 foot 6.5 inch, someone
Yeah.
can be 5 foot 7, 5 foot 8. All heights are possible, right?
Yes.
They said that what you did on pea plant does not make sense for humans. Pea is either tall or short, but humans are not either tall or short. They are everything in between, right?
Yeah.
Contemporaries could not prove it because you cannot do experiment on humans. But it was very easy to understand that by looking at humans around, they were not digesting the fact that alleles cannot blend, right?
Yeah.
So these were the two reasons, major reasons. There was another, write down. Thirdly, the third reason was, you know who was Mendel? Mendel is a very interesting person, like one of my favorite people from history of science. You know why? Because Mendel was not a biologist. Who was he? Mendel was a physicist.
Professor.
He was a physicist. He studied physics, he did physics, and he liked chemistry when he was in school. He said that, but he ended up doing physics. His profession was a priest. By profession, he was a priest in church. In earlier days, a lot of science was funded by church. You know why? Church used to believe
By looking at the Bible.
by
Looking at the Bible and making concepts.
yes, church used to believe that everything that science will prove will come closer to prove the existence of God.
Yeah.
That God is controlling this by this mechanism. So for church, science was the mechanism, underlying mechanism by which God works. Okay. So they used to fund this a lot. But then as science kept progressing, it started going against the church and against all religious sentiments, actually, where they started, Galileo came and started saying that, we are not at the center, like the Bible says. Earth is not at the center of the universe. We are revolving around the sun. Now, from there onwards, they started getting pissed off. But when Mendel started doing things, he was a priest. He got the support. By profession, he was a physicist, and he did biology. So when he did biology, botany, he used a lot of maths. Can you see all the crosses, the number, genotypic ratio, phenotypic ratio?
Yeah.
This was something new for biologists of that time. Biologists did not appreciate maths much at that time, and mathematicians did not appreciate biology much at that time. In today's time, we know that science is interdisciplinary. Everything is important for every other thing. But at that time, there were discrete branches. A lot of competition was there. So third reason was, write down, Mendel's approach used lots of mathematics to explain biological phenomena. Mendel's approach used mathematics to explain biological phenomena, which was totally new and unacceptable for the biologists at that time. Which was totally new and unacceptable. Okay. So his work remained hidden. No one paid attention to it. He said a lot of good things which could be proved mathematically about biology. No one paid attention. Because of this reason. Later on, in around 1900, so the real author for this theory comes. Okay, it was initially given by Mendel, but not paid attention to. But in around 1900, there was a scientist whose name was there were three scientists. Hugo de Vries, first was Hugo de Vries, second was Carl Correns, and third one was von Schermack. Von Schermack. Now this is a classic case that all of these three scientists were not working in a group, okay? All of them were present at different regions of the world. But around the same time, because this topic was very heated, people always used to discuss Mendel, some people used to disregard, some people used to accept. So during the 1900s, genetics was a hot topic in biology. And mathematicians were also interested because Mendel did it. He was a physicist. So these three scientists independently discovered this work of Mendel. Now, when Mendel wrote it, he wrote it as a book. Paragraph after paragraph, he's talking about things. You understand? Like a boring novel.
Yes.
No one had the patience, no biologist had the patience to sit, read, and understand. So they were like, oh, he has done some story thing. I did this, then 9:3:3:3. From 253 were this, 54 were this. So they did not pay attention. All these three scientists independently found the work and gave the work the status of a theory, the chromosomal theory of inheritance. So write down. The three scientists independently rediscovered Mendel's work. Mendel's work and raised it to the level of a acceptable theory. Raised it to a level of acceptable theory known as chromosomal theory of inheritance. And by that time, you could actually see chromosomes because chromosome staining was a technique invented. So when Mendel was there, he did not know of genes or how can we see chromosomes. But what he said, so who gave the theory? Who raised the laws to the status of theory? These three scientists. But who did the actual work for the theory?
Mendel.
Mendel.
Yes, perfect. Now write down what did the theory say? Write down. The chromosomal theory of inheritance states that there are discrete units which physically exist that gets inherited from one generation to another. That gets inherited from one generation to another. These were later found to be structures in the nucleus called chromosomes. Structures in the nucleus called chromosomes. Right? Chromosomes are physical things and they get inherited, right? From one generation to another. They are physical packets.
Yes.
Yes. Known as chromosomes. Write down one more point. Chromosomes replicate just before the cell division. Chromosomes replicate just before the cell division and are transferred to the next generation, next cell, right? Next generation cell. Now, there are certain things called homologous chromosomes and heterologous chromosomes. Okay. So let's come to chromosomes. Let's understand chromosomes a little bit. What do you know about chromosomes, Reem, already? I think Minashi knows slight more because I have taught. Let's come to you first. Reem, what are chromosomes?
They're present in nucleus,
Okay. Yes.
and nucleus is present in cells.
Okay.
Cells are present in our bodies.
What about chromosomes? Let's put the nucleus back. Now you are not allowed to go out of the nucleus. But inside the nucleus, what are they? You know what is a DNA?
Yeah.
Yes, what is a DNA?
DNA, it's our traits.
Mhm.
It decides our
Is DNA something physical or is it an idea?
it is physical.
It is physical. Like it's a long, it's a molecule, right?
Yeah.
But it's a very long molecule, right?
Yeah.
It's even longer than the length of cell. Sometimes, if you take the total DNA, all the DNA threads which are present in one haploid cell of our body, which is 23 DNA strands, right?
Yeah.
So those 23 DNA strands, if you open it up and align side by side, what will be the length?
No, what will be the length?
1.3 meters from one cell. Now, how do you think 1.3 meter long thread, very, very thin, is packed in a cell?
It is folded, I guess.
Yes, it is super coiled, not just folded. It is highly super coiled, and it is present in the form of chromosomes. Okay.
Okay.
So one chromosome is basically one long DNA thread, super coiled. So this is the DNA thread. Okay. If I open it, it will be very long, right?
Yeah.
This one super coils to become very, very densely super coiled to become this one thing called one chromosome. And this is a chromosome. Make sense?
Yeah.
But to coil something, you need a support, right?
Yeah.
Just to coil thread on a cardboard to make a spool, right? There has to be something on which you coil it, no? So those things are proteins, okay? So a chromosome contains DNA coiled over protein. Is it clear?
Yes, sir.
Okay. Now let's talk about some chromosomes. Now I will use, let's say, every chromosome has a structure like this. Okay. Now, all of this is actually coiled DNA, right? You understand, everyone?
Yes, sir.
It is all coiled form of DNA, super coiled. I'm still not making it super coiled. It is highly super coiled. Let me just fill this whole space. In a chromosome, this is chromosome number, let's say, 1. Chromosome 1. Okay. And this came from mother. Cool.
Yes.
In the center of a chromosome, there is a region which is known as Kinetochore. Kinetochore, some people call. It's called kinetochore. Okay. This is mostly protein structure. And every chromosome has a short arm and a long arm. The short arm is called P arm. Okay. The word P is for petite. What does the word petite means in English?
Skinny.
Tiny. Yes, you're right.
Yeah, skinny, small.
Small. Petite arm is the P arm. And the long arm is called Q arm. P and Q. Is it clear?
Yes, sir.
Very good. Now this is chromosome number 1 as it got transferred from one parent. Now let me make another chromosome number 1. Okay. I'm just coloring it different to explain you certain concepts. Okay, and please pay attention to these concepts. They are not in NCERT in a proper detail. You will be confused from NCERT a lot. So understand it here. So this is the chromosome which came from mother, like from the ovum. Okay.
Okay, sir.
And the same, this is chromosome number 1. It came from the sperm, which is from the male. Clear? But both of them are same chromosomes, like same in the sense they are the same thing. Like it has a kinetochore as well, right? And it has
Yes.
a arm and it has a Q arm. I think the Q arm is not proper long. Can you figure out which is the long one?
Yeah.
We measure it from the kinetochore. Now in this 2 chromosome, I'm using a red ink. So red. What will be a good contrasting color for both red and blue? Yellow?
Black.
Black? Or yellow? Let me go for yellow. Let's see. We will change. It does not.
Okay.
Now, on these 2 chromosome number 1, both have a short arm. Both have a long arm. And both have a kinetochore. These are called, these chromosomes come like this. But just before the cell division, they replicate. Okay. And when they replicate, the whole chromosome looks like this. You understand?
Yes, sir.
Because what is happening there? They have replicated. Yes. I will just keep it unshaded. Now, pay attention. These 2 chromosomes, which are the same chromosome pair but from different parents, are called homologous chromosomes. What are they called?
Homologous chromosomes.
Yes. Is it clear?
Yes, sir.
In the same chromosome, this one is called a chromatid. What is it called?
Chromatid.
Before, in the resting phase, when like the chromosome is packed and sent, there is only one single chromatid like this. Okay, like this pen. But just before cell division is going to happen, because when cells will divide, you have to send equal genetic material, right? Remember replication?
Yes.
So just before the cell division, there are two chromatids formed. Okay. And both of these are called sister chromatids. What are they called?
Sister chromatids.
Sister chromatids are present on the same chromosome. Remember this. If I take one chromatid from here and one from here, they will be not be called sister. They will be called non-sister chromatids. Is it clear, everyone?
Yes, sir.
Same chromosome, when it replicates, it forms two sister chromatids. But chromatid from one homologous and other homologous are called non-sister chromatids. Is it clear?
Yes, sir.
Okay, very good. So, and heterologous chromosome is one is chromosome number 1, the another one is chromosome number 3. You understand? Not related. They are heterologous. That is not necessary for genetics. In genetics, you have to remember what is a homologous chromosome. They come, the same chromosome come from different sex. And what are sister chromatids? Chromatid which are formed after replication of the same chromosome. And what are non-sister? The different chromatids of the same homologous chromosome. Is it clear?
Yes, sir.
We will not call two chromatids non-sister if one is from chromosome 1 and one is from chromosome 3. The concept of sister or non-sister only happens in homologous. If it is a heterologous chromosome, it is of no use for genetics. Forget about it. Is it clear, everyone? It has some use, but I'm not going to complicate things here. Is this simple thing clear?
Yes, sir.
Very good. So just make this diagram first, then I'll move forward. Homologous chromosomes always have the same length. Okay, keep writing certainly. Have you drawn this, both of you? No?
No, sir, I'm drawing.
Please draw and label everything very carefully. If you understand this, then linkage recombination becomes very, very easy, because always these terms comes: sister chromatid, non-sister chromatid, gene, allele, locus. Take your time, tell me when you are done.
Done, sir.
Okay. So if you see, when this chromosome had not replicated, right? And this chromosome has also not replicated to form sister chromatids. They basically, the chromosome itself is the non-sister chromatids, yes or no?
Yes.
Before it has replicated, it is this one single and this one single, and they are called non-sister chromatids. So non-sister chromatids are same as homologous chromosomes, yes or no?
Yes.
Yes. Write down. This is a neat question. Which of the following is same as homologous chromosomes? Sister chromatids, non-sister chromatids, heterologous chromosomes, none of the above. And students always choose none of the above because they think chromatid cannot be chromosomes and heterologous cannot it be the right option. But non-sister chromatid is actually homologous chromosomes to begin with. Then these homologous chromosomes replicate and form two sister chromatids each. Yes?
Yes.
Very good. Now, let me take a look. If there is a Write down this one line. Homologous chromosome pairs have the same length, same staining pattern. When you stain a chromosome with a color, with a dye, they have the same staining pattern, same centromere position. The nitochore has the centromere. This is centromere, okay, where the nitochore is present. Is it clear, everyone?
Yes, sir.
Centromere position or nitochore is the same thing. Nitochore is the name given to it, and the position is centromere. They have the same centromere position, same length of P and Q arm, same length of P and Q arm, comma, same characteristics of the genes. Same genes are present. If chromosome 1 from female have 20 genes, this will also have the same 20 genes. So same characteristics of the genes, and also same location of the gene on the chromosome. Same location of the gene on the chromosome. And it is called a locus. So I am drawing gene 1 here. So let's say this is gene. Is this visible to you, all of you? This yellow mark?
Yes, sir.
Let's say this is gene A. Okay? This is a allele. Right? You understand? This is allele for gene A. Then the same gene, but a different or the same allele will be present at the same position in a homologous chromosome. Is this also visible?
Yes, sir.
So I'll appreciate if you use another color marker for your notes also. And this is also gene A. And their position is called locus. So they have same locus. Every gene has a same locus. Locus means location on homologous chromosomes. Okay. Just to give some more clarity, let me use green color as well and make another gene. If this is gene B, and here also at the same position will be gene B. Is it clear?
Yes, sir.
And now if sister chromatids are replicated forms, then clearly they will also have the same location of gene A and gene B, because this is easy to understand. They have just replicated, right? Copy. Sister chromatids are copy of each other. Make sense?
Yes, sir.
So in a way, all these 4 chromatids will have the same location, everything, right? Only the alleles will be different. So this might be, let's say gene A is for tallness. So this might contain capital T, but this might contain small t. But the same gene, different allele at the same location. Is it clear?
Yes, sir.
And this is how, when both of these chromosomes come together in a new next generation, if there is a dominant allele, that is expresses over the recessive allele. Make sense, people?
Yes, sir.
Draw this also. Write down non-sister chromatids. Non-sister chromatids. I'm using this space here. non-sister chromatids are formed non-sister chromatids are formed during meiosis division. Okay, clear?
Okay, sir.
Perfect. Okay. Now write down, there are two scientists named, very important, Sutton and Boveri. Sutton and Boveri. These are the two scientists that collated and called this theory as chromosomal theory of inheritance. So write down, Sutton and Boveri said or stated, they stated that pairing and separation of a pair of chromosome. Remember, when I'm saying pair of chromosome, I'm talking about homologous chromosomes. Okay, clear?
Yes, sir.
So
Yes,
pairing
sir.
separation of a pair of chromosomes would lead to the segregation of a pair of genes. You can also write factors, because at that time it was known as factors. A pair of genes or factors that they carry. Okay, write down, this is known as chromosomal theory of inheritance. Now let me give you three questions. All three are confusing questions, but has come in entrance. If the question is, who gave the chromosomal theory of inheritance, then it is Sutton and Boveri, because they finalized, clubbed everything and gave it as a theory. If the question is, who raised Mendel's work to the status of a law, then it is these three. I don't make this error, it's confusing. Write down these three raised the status of Mendel's work to laws. Okay, because Mendel wrote a book like a novel, but who did all the work? Mendel. Okay.
Okay, sir.
Okay. So Sutton and Boveri, remember that. So now we have to understand linkage and recombination. Are things clear till here? No doubt.
Yes, sir.
Now write down. Shall I? Write down linkage. Let's go with linkage. I was thinking which will be the easier way to go. Okay, I'll bring that concept later on. Write down linkage. Linkage of genes. Since you have understood the structure and how a chromosome looks like, where are the genes present, they are called locus. Now linkage of genes is actually a physical linkage. Like these genes are present so close to each other that the chances of these genes going forward in the same together is more. Okay. Now what did I say to make it easier? Write down. Who figured out linkage? Write down. Morgan. The scientist's name is Morgan. Morgan discovered linkage of certain genes in Drosophila. You know what is a Drosophila? It is a fruit fly. If you keep fruit or fruit peels somewhere in your house or in the dustbin, you will see near that fruit there will be very small flies hovering. Sometimes we misunderstand them as baby house flies. They are not house flies. If you look carefully, they have red eyes. They are called fruit flies. Okay. Drosophila. Fruit flies are very famous model system for research in science, especially in genetics. So Morgan discovered the linkage of certain genes in Drosophila. Now, by this he meant that write down. Morgan crossed yellow-bodied white-eyed females to brown-bodied red-eyed male. Okay. This was the actual cross. This can be asked in entrance, so that's why I'm explaining it to you. Now let's say yellow body is one character, just like round seed and yellow seed, right?
Yes, sir.
White eye is another character, right? Just like round seed and yellow seed. Seed color, seed shape. Or let's say round seed and tall plant. Clear? You understand?
Yes, sir.
There, in that case, following the law of independent assortment, both the characters will assort independent of each other and all possible combinations will be formed. Yes or no?
Yes.
You will expect that in the F2 progeny, there will be yellow bodied with red eye, yellow bodied with white eye, brown bodied with red eye, and brown bodied with white eye. Right? This is what you will expect normally by laws of Mendel.
Yes.
But here it did not happen. So write down, when this cross was made, he observed that the two characters did not segregate independent of each other. The two characters or genes, you can write genes or you can write characters. The two genes, write genes, because now we are talking about chromosome and genes in that language. So the two genes did not segregate independent of each other. If they would have segregated independent of each other, what would have been the ratio, phenotypic ratio? F2.
9:3:3:1.
Yes. So this has to be followed, right? But this was not followed. Okay. So write down. The two genes did not segregate independent of each other. And in the F2 generation, the phenotypic ratio was drastically different from 9:3:3:1 ratio. He found that majority of flies were yellow bodied and white eyed only. Okay. So how many flies should have been yellow bodied and white eyed? Let's say if yellow body color is dominant and white eye is dominant, let's say, then 9 should be this, 3 should be this, other 3 should be a combination, and 1 should be recessive for both. Yes.
Yes.
Yeah. But he found that around 70 to 80% of the progenies were just like parents, as if no segregation happened, or 80% of the time there was no segregation. Still 20% of the time there was segregation. So that is a deviation to the third law, and it is called linkage of genes. So write down. Morgan figured out that there was a physical association. So these two genes were linked. Okay, these two genes were linked. There was a physical association or linkage present between the two genes. Okay. That's why no different combinations from the parents, or very few different combinations from the parents were possible. Combination is only possible when these two genes are independent, right? It's like if Reem and Minashi every time goes together to parties, then we will only find them together in each party, right? In some,
Yes.
if Reem is not present, then Minashi is also not present. So this is called linkage of genes. Is this concept of linkage clear to you? And you know,
Yes, sir.
why does this happen? Because this happens because of their physical presence. So if gene A is present here and if gene B is present here, let me make it here. Let's say this is you understand, this is a chromosome. Okay. If this is gene A and this is gene B, correct? Then the chances of these two segregating is more because the distance is more, right? Now I will teach you about recombination next and we'll understand. But if there is a gene called, let's say, C, which is present just adjacent to A, you understand? Then
Yes,
A
sir.
tends to be linked. So they have more linkage in between. Is it clear? Whereas these two
Yes,
genes,
sir.
linkage. Now, before going forward, let me also tell you what is. So linkage concept is clear.
Yes, sir.
Now let's come to recombination. Write down recombination. Recombination of genes. Okay. Am I audible of you? Hello.
So,
Yes, sir.
Okay. Write down. Recombination means recombination means the rearrangement. You can say gene or DNA both. Recombination means the rearrangement of the DNA sequences by the breakage, by the physical breakage and rejoining of chromosomes of chromosomes or segments of chromosomes or segments of chromosome. Now there is a figure given in your NCRT. Can you come back? All of you have, both of you have NCRT with you right now? There's a
sir.
very,
No, sir. I have to go and take.
can you go and fetch it, Reem?
Yeah.
Is it there with you, right? Can you take it? Yes. Please open it. Minashi, you also. You have NCRT with you?
Yes, sir.
Yeah. Okay. People, are you back? Both of you with book?
Yes, sir.
Yes. So tell me, no? Now, I'm going to tell you some higher concepts now. You have to pay attention and ask me at any step where you are confused. Please open page number 84 and keep it in front of you. Before understanding what happens in page number 84, we have to understand another concept which is not clearly given here. So I will be drawing, okay? Now, both of you open page number 84. Can you see figure 5.11?
Yes, sir.
Yes.
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 Now write down. Before that, keep it in front of you and look at the screen. Chromosomes, basically during the meiosis, when they are making gametes, they undergo a event called crossing over event. So write down what is question. Write down question. What is crossing over of chromosomes? Crossing over of chromosomes. And to explain this, I will go to the same old these two chromosomes. Okay. Remember the color? Now
Yes,
let's
sir.
come to the same chromosome. So let's assume that there are two same chromosomes. Now I will make both. Okay. And in the center, what is there? Centromere, correct?
Yes.
This is the chromosome from the, you will also color it. I will tell you why it is important. So let's say this is chromosome number 1 from the mother, from the female, okay? Same color coding which we just did above. You also please use the same colors if you are using colors. You have to use colors actually. In this diagram, you have to use colors. Okay, clear? Now let's make
Yes,
the,
sir.
let's first write. So this is chromosome number 1 from the female. And let's say try to make it as similar as you can, but you know, you will never be able to make it exactly similar. That's one limitation of hand drawing. I hope I'll make it similar. Now this is the homologous chromosome, same chromosome number 1 from father. Is it clear?
Yes, sir.
I have to do some hard work here, but let's do it for clarity. Because this is a very, very confusing concept if not understood properly. Okay, everyone? It also has the centromere at the same position. This is chromosome number 1 from the father, which makes both of them what does it makes both of them? What are both of these?
Homologous chromosomes.
Crossing over happens in homologous chromosomes only. First thing you understood? It happens in homologous chromosome only. Okay. Now, what happens during chromosomes? Let's also mark these two are homologous pairs, and now let's also mark what are these two? These two?
Sister chromatids.
Yes, these are sister chromatids. I have to write it like this. And these are also sister chromatids, right? And if I talk about these two, what are these two?
Non-sister chromatids.
Yes. So I am removing this part, okay? I'm deleting this part. I need this space. Can I delete this part?
Yes, sir.
These are non-sister. Okay, clear everyone?
Yes.
Okay, perfect. Now what happens is during crossing over, these two are present together, okay? Whenever they come together, okay, from different parents, now haploid is becoming diploid. So what happens is they exchange genetic material. Homologous chromosomes exchange genetic material between non-sister chromatids. Write down this statement. Homologous chromosomes exchange genetic material between non-sister chromatids. Now what does this mean is, I will draw it something like this. Now they come very close. In this leg of the chromosome merges with make sense?
Yes, sir.
We will not do this in hurry. We will do this beautifully. Important concept. I hope both of you are using different colors. Are you?
I am.
Nashi?
Yes, sir.
Yes. You're using two different colors, right?
Yes.
This is art and science. We're having an art class today with concepts of science. I could have used a picture to show you this, but I think this is more, this stays in your head when you make it yourself, right?
Yes.
Yes. Now, at this point where this crossing over is happening, these two are sister chromatids or non-sister chromatids?
Non-sister.
Non-sister.
These are sister. And when they cross over each other, there is a physical break, and the physical break happens here. At this junction, there is a physical break, and because of this physical break, you get a combination, a different rearrangement of DNA. Make it here. Because of this, we get this. Okay, carefully, let's draw this. So we get this. Are you understanding, both of you?
Yes.
Oh, just wait, the cursor is not picking up right. Chromosome that came from one sex, one source, was having certain genes, no?
Yes.
And the chromosome that came from the other parent was having some other alleles for the same genes, right? It could be the same
Yes.
genes, or it could be the different alleles. You understand? But when both of them underwent meiosis, this happened. They did a crossover at one junction. Now this crossover can happen anywhere, on P or on Q, anywhere. I have just shown it this way. It's not like always this side, it happens like this. I hope that this is clear, right? But
Yes,
crossover
sir.
happens between chromosomes. And when it happens, physically, one segment of that chromosome breaks and attaches itself to the other. So there is an exchange. Still the length remains the same, right? You see the chromosomes both are the same yet, but the difference is just one moment. This is the centromere, this is the centromere, this is the centromere, this is the centromere. Now, can you see that after the crossover point, one part, this crossover point is called chiasmata. What it is called? Chiasmata. Okay, the crossing over, overlap things. In this phase, exchange of genetic material happens between two chromosomes. Okay, and finally, here, these two chromosomes are called recombinant chromatids. What are they called? they have undergone recombination. Is it clear?
Yes, sir.
And
Yes, sir.
these two chromatids are called non-recombinant chromatids. Is it clear? But both recombinant
Yes, sir.
chromatids and non-recombinant chromatids both are non-sister chromatids. Is it clear? Yes or no?
Yes.
These two are also non-sister and these two are also non-sister, right? Do you understand, everyone?
Yes, sir.
Reem?
Yes, sir.
So write down recombinant chromatids and non-recombinant chromatids both are non-sister chromatids. Both are non-sister chromatids. But let's see what happened basically. Now, since this is in front of us, we will understand what is a crossing over and how it gives rise to recombinations. Now, I will use, let's say there was, let's say 3 genes. Not selecting, let's wait people. Let's say I'll use the color light green. There is one gene present here, okay? So it will also be present here. Am I correct? And it will also be present here, right?
Yes.
This is A. And let's say there are 2 genes which are, one is present here. Can you see this yellow mark, everyone? Is this yellow visible or not?
Yes, sir. It is visible.
Okay, good. Let's say this is B. Let's use some other color contrasting one. Which color should I use? Let me use white. Let's see if white works. White is not being selected. Some issues. Yes, white is selected. Let's say there is another gene very close here. Can you see this white mark, everyone? Both of you? Oh, but how will I?
Yes, sir.
Oh, let's not take white. Background is white, white. No, I cannot write with white. Let's say black only. Can you see this third gene? People say yes or no. This is gene.
Yes, sir.
So these 3 are our genes of interest. Now these 3 genes are present here. In this case, when they are making a crossover, gene A will have no effect, right? Yes.
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 But about B, let me write about alleles. So I will use, let's say I will use color green first. If it is gene A, then for gene A, let's say this is the gene for tallness. So this will have capital T and capital T. And let's say this has small t and small t. Is it clear?
Yes, sir.
And similarly, for gene B, let's say gene B is for color of the seed. So this will be capital yellow, capital Y and capital Y, right? And this will be small y and small y. Is it clear?
Yes, sir.
Yes, sir.
And let's say the third one is for what? Tall white, let's say wrinkled and this was yellow and green, round and wrinkled. So let's say this is capital R, and this is small r, right? Because these are two, these will be same because they are duplicates of the same. So sister chromatids are always duplicates, right? That
Yes.
when this will happen and we will reach this stage, you can now understand, and I hope you understand that capital T will stay unchanged, right? This chromatid will have a capital T. This will also have a capital T. Is it clear?
Yes, sir.
This here will have a small t. And this will also have a small t. Perfect. Now, let's look here what happened during the in-between. So in between, the gene for tallness is sorted. It is not undergoing crossover, so it will stay the same. Now if the crossover position, this bridge where it is, if Y, like for yellow and green, the gene yellow was above the bridge, okay? Clear?
Yes.
Capital Y, capital Y. This was also above. This will be small y and small y. Clear?
Yes, sir.
But the gene for round was below. This here is capital R and capital R. And in between yellow and round, like these two genes, there was a crossing over event. So the small r is somewhere here, right? Hidden beneath. You understood?
Yes.
It's not visible, but I'm marking it here. Small r. Now, when the crossover happened, you will see that we will get all different permutation combinations. First, there will be capital Y, yes. Here also there will be capital Y, yes. Here it will be small y, and here it will be small y. Correct, everyone? This is
Yes, sir.
because of crossing over. Tell me what will be the gene present on this chromatid? Capital R. But what will be the gene present on this chromatid?
Smaller.
Small r. And here?
Capital R.
Capital R. And here?
Smaller.
Now there are 4 chromatids possible. Now when they will divide each, there will be from one meiosis 4 gametes are formed, right? So this will split from the center and this will split from the center, and there will be 4 chromatids, right?
Yes, sir.
Let's say this is chromatid 1, this is chromatid 2, this is chromatid 3, and this is chromatid 4 after they are all separated. So for this one, what will be the gene combination? Capital T, capital Y, and capital R, right?
Yes.
For the 4th one, the gene combination will be small t,
Small y and small r.
small y. These two are like parents only, right?
Yeah.
Non-recombinants. Do you understand why we call it non-recombinant now?
Yes.
But for second one, I'm writing recombinants with another color. Let me use blue color. You will figure out for the second one, the gene combination will be capital T, capital Y, and small r. Something which was not present in the parents, right?
Yes.
And for third chromatid, it will be small t, small y, and capital R. Do you understand? These four are different types of genotypes. Yes or no? Recombinant.
Yes, sir.
So if these two, if 1 and 4, if 1 and 4 goes to a progeny, they will have the same set of characters as of parents. Yes, 9:3. But if 3 and 4 chromatid goes to a gamete, like to one pair, one offspring, then they will have, sorry, this is not 3, this is 2. Oh. But if 2 and 3 goes to a progeny, they will be having different characters than from the parents. Do you understand?
Yes, sir.
This whole drawing making sense to you? This whole half an hour was for you to understand what is crossing over and how are recombinants formed. Yes?
Yes.
Please draw this. Take 5 minutes. Please mark everything correct, draw it, and let me know. Then I will tell you about linkage, how linkage works in the same diagram. Okay. And also write these 4 different types of combinations, recombinants. So 2 and 3 are recombinant chromatids, and 1 and 4 are non-recombinant chromatids. Let me know when you are done. Take 2-3 minutes. Do it without any hassle. Those who have missed today's class, Reem, tomorrow you are going to give a recap to them. Please.
Okay, sir.
Okay. I pass on this burden to you. Are you cursing right now? Me for this.
No, sir.
I first decided to use an image and get it through. But then I thought, students struggle. Unless they do it themselves, they struggle. How did recombinants came?
Done, sir.
Done? Manashi,
Yes, sir. Done.
you're doing or you're done?
Done, sir.
Done. Okay, perfect. Good. Now pay attention here. Did you notice that recombination happened between Y and R, the yellow gene and the black gene? Yes?
Yes.
And why so? Because the crossing over happened between these two genes, right?
Yes.
If crossing over would have happened between T and Y here, somewhere here, then recombination would have been between T and Y, right? The green gene and yellow gene. So the region, the location where the crossing over takes place. Now crossing over can happen anywhere on the chromosome. I have shown here, it can happen between these two non-sister chromatids. It can happen between these two. It can happen between these two only, but at a different location. Do you understand? Anywhere. At the same time, two crossing overs can also happen, like one in the Q arm, one in the P arm. Let's say there was a fourth gene also, and in between the green and let's say there was a another gene here, maroon gene. Between green and maroon also, if there was a recombination, then they will also be forming recombinants. Yes.
Yes.
Now, having said that, it is very clear that if you have two, and this recombination event is random, there are various hotspots, there are various weak points in the chromosome which are preferred, but they can happen anywhere. But some regions are more preferred for recombination, and those regions which are little far from the centromere, from the kinetochore. Is it clear, everyone? Those are more.
Yes, sir.
So if two genes are more closer to each other, for example, with this R, with this black gene, if there was another gene, let's say sky blue in color, I'm making a sky blue gene. If there was another gene here, very close to it, okay? You understand? Very close to it here. So when this crossover happened, the chances of they getting segregated would have been more or less?
More.
Less. The closer the genes are, the chances, say this is my finger, okay? On my finger, you see these markings?
Yes.
Very good. Now let me use a pen to make more markings. Oh, I'm so sorry. Am I still audible or am I not audible?
You are.
And I'm visible also now, right?
Yeah.
So let me mark. Let me make it on this one. The pen is not working. What a good. This one I have. There's a third mark I made. Can everyone see this? Let's see, this is a chromosome. There is already a gene here, which is my natural curve. Can you see the natural mark?
Yes, sir.
Gene A, gene B, gene C. Okay. Now, if I ask you randomly to cut, like slice my finger randomly, then randomly the chances are it can slice from here, right?
Yeah.
It can slice from here. It can slice from here. But what are the chances that there is very less space between A and B and it can get sliced from here? What are the chances? Very high or very low?
Low.
Low.
Very low, right? Because if I randomly slice, it can hit. So the space here and space here is very large, right?
Yeah.
These two genes are very close together on the chromosome. Like genes which have very close locus to each other, they have more linkage and less recombination. Is it clear?
Yeah.
And those genes which are far away from each other, let's say the black one and the green one here. So you will see that if I randomly slice, why I'm using my finger now? Let me use here. Yes, can you see the cursor, everyone? This green gene and this black gene, they are very far away, right? On extreme ends. So anywhere if recombination happens, there are chances that these two genes will be segregated all the time, always, right?
Yeah.
Only if slicing happens here, then these two genes will go together. Yes or no?
Yes.
So the farther away the two genes are present on the chromosome, the farther away their locus is, less is the chances of linkage and more is the chances of recombination. Sorry, it got stopped, right? Am I audible and visible still, Minashi and Reem?
Yes, sir.
Yes, sir.
By the time it's opening again. Do you understand what I meant to say?
Yes, sir.
Closer the genes are present. Yes, it's open. Let me share it. Yes. Can you see the screen again?
Yes, sir.
Closer the genes are present on a chromosome, lesser is the chance of their recombination and greater is the chance of their linkage. Okay.
Okay, sir.
So write down. You have drawn this, write down. Now come to the figure in the end. Come to page number 84. Now you will understand that figure. Do you see cross A and cross B? Everyone?
Yes, sir.
In cross A, look carefully. Do you see that the female has 2 chromosomes, the male has just 1, and there is one wild type? Like, sorry, one is blue. Do you see the blue one, which is curved,
Yeah.
tilted, curved? So that is the Y chromosome. Okay, they have taken X chromosome here into consideration. So males only have 1X, females have 2X, right? So you see in female, both the chromosomes have a centromere. Can you see at the very end, towards the very end, there is a centromere? White.
Yeah, sir.
Yeah, that is the centromere. It is not in the center, it's towards the end. These type of chromosomes are called telocentric chromosomes. But you see that on yellow, which is the female chromosomes, there is 2 genes, one for yellow body and one for white eye, Y and W. Do you see that?
Yeah.
The same different alleles for the same gene is present in male also, in the blue chromosome, Y plus and W plus. Yes?
Yes, sir.
Yes.
Which is for, if the female is for yellow and white, the blue Y plus is for
Male.
brown and red. Okay, you understand? Y plus is for brown and W plus is for red, because the other allele for white is red and the other allele for yellow is brown. Yes.
Yes, sir.
Yes, sir.
NCERT that they have not mentioned everything clearly, so people get very confused, but you can understand now. We are crossing between the two. Okay, the male and female. Now look at F1 generation, cross A. Don't go to cross B again. Look at cross A F1 generation. Can you see that in F1 generation, the chromosome got switched? The blue one came in female and the yellow one went to the male. Can you see that?
Yeah.
Changes happened. Now, pay attention that now these are 2 homologous chromosomes. Yes or no?
Yes.
Yes, yellow and blue are homologous chromosomes.
Yeah.
One containing Y, yellow has Y and W, and blue has Y plus and W plus. Just like I simplified it through R and small r and Y and small y. They are just saying Y and Y plus. Same concept. Are you understanding?
Yes, sir.
Now in cross A, when you go from F1 to F2, there is a meiosis happening and recombination happening. Can you see in F2 generation, there are 4 progenies possible. Can you see in progeny number, like in 2 of the progenies, there is recombination already happened? See why a slight blue is present with yellow in 2 of the cases. Can you see that?
Yes, sir.
And they are called recombinant types, and they are just 1.3%. If recombinants are 1.3%, parental will be 98.7%. What does this tell you about these 2 genes, Y and W? This tells that between the yellow body color and white eye color, there is lots of linkage. These 2 genes are very close to each other. That's why 98.7% of the time, the parental type is only present, but the recombinants are very less, 1.3%. Do you understand?
Yes, sir.
Simple. That's all you had to understand in this. Now let's come to cross B. That is a case of 2 different genes. Again, it is for W and M. W is for white eye and M is for miniature wings. Okay. Can you see white and miniature written?
Yes, sir.
Can you
Yes,
also
sir.
see the distance between the two is more than the distance was between Y and W? Pay attention.
Yeah.
Yeah. If the distance is more, now following the same thing, in the F2 generation, parental type is how much? 62.8%, right?
Yeah.
Is it more or less than the first cross?
It's less.
Yes. And recombinant types are 37.2%, which is more than the cross A, right?
Yeah.
What does this tell you about these two genes, white and miniature? Are they linked? No.
No.
Less linked than white, yellow and white. White and miniature are less linked characters than white and yellow. That's why recombinant types are more here. 37% of the progenies are recombinant, which means almost one, more than 1 in 3. Yes, in every 3, 1 will be a recombinant. On the other side, in every 100, only 1 or 2 will be recombinant. Yes,
Yes.
very good. So this graph was very, very complicated to begin with. That's why I focused on this one first. Okay, is it clear to both of you? Both
Yes, sir.
combination. Now the rule is, write down the rule here. Linkage between any gene is inversely proportional to recombination. You know about proportionality from physics? This is the formula. Do you understand, everyone?
Yes, sir.
What does this means? More linkage,
Yes, recombination.
less recombinants. Okay, and less linkage, more recombinants. Is it clear?
Yes, sir.
And the factor on which they depend, write down, is linkage depends on the physical location or physical proximity. Proximity means closeness, right? Of two genes. Genes which are closer to each other are tend to be more linked. Genes which are farther away from each other are tend to be more recombinant, less linked. Okay? Yes or no?
Yes, sir.
Now, when Mendel took all those 7 traits that we studied in the beginning, you all were in the class when we studied in the beginning? Mendel
Yes.
7 traits, these 7. And he did not find linkage, which means by just sheer luck, all these genes were very distant from each other on the chromosomes, on different chromosomes. Yes.
Yeah.
So he just could not find the 2 genes. Otherwise, Mendel would not have given the law of, like, would not have written about it as a law of independent assortment. But later on, people found in Drosophila, Morgan found in Drosophila, that all the genes are not independently segregated, and some genes are linked together because they are very close to each other on the same chromosome. So if a question comes that why could not Mendel found linkage, there are 2 reasons for it. Either if 2 genes were present on the same chromosome, they were very distant from each other, or all these genes were present on different chromosomes. Yes.
Yes, sir.
So it's a sheer luck, but science ultimately over time unfolds and the truth comes out. So the truth is that law of independent assortment is not a universal law. It has linkage and recombination as exceptions. Okay, people.
Okay, sir.
Okay, so this is where we will end today. Please go back, revise linkage recombination from NCERT, and we will spend some more time on this, and then we will go to other exceptions like polygenic inheritance, pleiotropy, and finally sex determination in different organisms. Okay.
Okay, sir.
And then we also have to go to mutation, then genetic disorders. It is a very long chapter, I can see. Very good. Genetics as a top unit is a very, very concept heavy unit. So pay attention. If you are going sequence wise with me, story will be in your head. Any doubt till now from day 1 to now of genetics?
No, sir.
Minashi, all good, all clear?
Yeah.
Yes.
Okay, very good. So see you both tomorrow. And Reem and Minashi, please help me with the recap for others who will join tomorrow.
Okay, sir.
Did
Thank you.